Hello and welcome to the next episode of the Psychedelic Medicine Podcast. I'm Dr. Lynn Marie Morsky, your guide on this journey, and today we're going to be discussing the intersection of psychedelics and the gut microbiome and the implications that may have for future personalized medicine with Dr. Giorgia Caspani. Giorgia is a neuroscientist and computational biologist working at the intersection of gut microbiome, psychedelics, and personalized medicine. Now, before we get to the episode, just a reminder that the Psychedelic Medicine Podcast is for educational and informational purposes only. Nothing here is to be construed as medical or legal advice. And one more reminder that if you or someone you know is a clinician and you'd like to learn more about psychedelic medicine, then please check out the Psychedelic Medicine Association. It is our mission to get clinicians of all types, therapists, social workers, doctors, nurses, up to speed so that you feel comfortable having discussions about psychedelics with your patients. You can find us at psychedelicmedicineassociation.org. Now, without further ado, thanks so much for joining us today, Giorgia.
Hey Marie, thank you for having me. It's a pleasure to be here.
I'm really excited I was excited when I saw your article that you were part of the team that wrote this article about the intersection of psychedelics and the gut microbiome, because personalized medicine, which if people don't know, it is tailoring medicine specifically to somebody based on perhaps their DNA or the gut microbiome or other aspects of that person, is so important in doing things like minimizing side effects and maximizing effect. And so when I saw you guys were focused on this, I was very excited. Can you talk a little bit? By the way, I'm just going to read off the title of your paper and we'll put a link to it in the show notes. But if anybody wants to follow along, the paper title is Mind Over Matter, the Microbial Mindscapes of Psychedelics and the Gut-Brain Axis. So let's start by having you explain to people, what is the gut-brain axis?
Absolutely. So the gut-brain axis refers, as the name implies, to a bidirectional communication network between the gastrointestinal tract and the central nervous system. And a key player in the gut-brain axis is the gut microbiota. Now, the gut microbiota refers to the ensemble of microorganisms from bacteria, archaea, fungi, viruses that reside in the gastrointestinal tract. And what I always like to mention when talking about the gut microbiome or the gut microbiota is the fact that humans and microbes have co-evolved for pretty much since their existence, so for hundreds of thousands of years. And so over this prolonged period of time of co-evolution, they have this symbiotic relationship has emerged, which means that they now shape their respective physiologies, right? So without our gut microbes, we wouldn't be able to synthesize vitamins or other bioactive compounds. We wouldn't be able to digest certain foods. We wouldn't have such a strong immune system either, because our gut microbes are essential for priming our immune system in early life. So they basically train our immune system to be able to discriminate between pathogens in our gut or in our body versus commensal bacteria that are actually beneficial to us. And also another fun fact that I always like to mention is that the gut microbiome, or the genetic information that is contained in the microbiome, this is known as metagenome, actually exceeds the genetic information present in the human cells by approximately 100 to 150 times, which means that some of the information encoded by this metagenome is exclusive to gut microbes, which means that without our gut microbes, as I said, we wouldn't be able to really perform a lot of the metabolic functions that we rely on as humans. And so going back to what the gut microbiome and gut-brain axis is this bidirectional link between our gut and the brain. It explains how, for example, our brain and our psychological state can have an impact on gut motility or gut secretions. Every time we feel stressed or anxious, we do feel that in our gut, right, in our belly. But also this is the top-down effect, but also bottom up. So it also explains how our gut microbes can influence the way we feel. And an example of that is how the composition of the gut microbiota and the secretion of gut hormones and so on can actually shape our sense of appetite. They have a strong effect on how we're feeling, our moods, our depression or anxiety, etc. So there's different pathways, biological pathways that explain how gut microbes can communicate with the brain and vice versa. We have neural, immune, hormonal, and metabolic pathways. But I'm happy to delve more in details into them. But this is just a general review as to how gut microbes can impact our brain.
Very cool. I am so like on the edge of my seat because I would love for you to dive into all those. But I will ask before, to keep on topic with our lovely show here is that do we know what changes psychedelics produce in the gut microbiome?
Excellent question. So this is pretty much what my review paper was about. So you're all welcome to read it. The spoiler here is that we actually don't know so much about it. Up until now, there's only one paper that has been published on the direct effect that psychedelic drugs have on the microbiome. This was a paper that investigated the effect of two psychedelic drugs, psilocybin and an analog of psilocybin called norbicistin, on the composition of the gut microbiome in rats. So these are rats that were treated with these two drugs, and then the gut microbiome composition was assessed and measured. And what the authors found was that there wasn't actually a difference in the overall microbial composition. So in terms of what in the field we call alpha diversity, so the richness and the evenness of the microbial communities, that was not affected. However, the relative abundance of certain species and bacterial taxa was affected. So depending on the drug, depending on the dose and the time interval, different bacterial taxa were affected, suggesting that yes, the psychedelic drugs can have an effect on the gut microbiome. However, this is very preliminary research. The sample size is also quite limited. So because of the experimental design itself, these results are not really generalizable yet. So we can't really draw much of a conclusion. And further studies are needed to really answer this question. However, if you read my review paper, you will see that I collected evidence primarily based on the similarity between psychedelic drugs and serotonin to describe my rationale, explain my rationale as to why I think that psychedelics can have an impact on the gut microbiome.
And as a follow-up, as you discussed, everything is bidirectional. How does the gut microbiome affect how drugs, including psychedelics, are metabolized?
Yeah, another excellent question. So we do know that the composition of our gut microbiome has a direct effect on how pharmacological drugs are metabolized. Gut microbes can directly metabolize drugs that are taken orally. Once in the gastrointestinal tract, they can either activate them into their active forms, they can break them down into their inactive forms. They can even create and form toxic byproducts. So it is essential to know how the gut microbiome metabolizes drugs and how it shapes and modulates the bioavailability of these drugs. Just to give you an example, we do know that the composition of the gut microbiome shapes responses to antidepressant drugs. For example, SSRIs, so selective serotonin reuptake inhibitors. We know that there's a correlation, a direct correlation between certain bacterial species and the efficacy of SSRIs in depressed patients. So definitely, yes, the gut microbiome can metabolize drugs. It's just a matter of more research being carried out onto exactly how and which microbes are responsible for the breaking down and metabolism of psychedelic drugs in the gut.
Yeah, I'm so fascinated by that. I did not know that we already knew that about SSRIs. So that's very interesting. You had talked about how there were like the four mechanisms by which the gut-brain axis affects, let's say, our well, how it communicates. And one of the topics you had mentioned when you were discussing that was stress and mood. Can you talk a little bit about how the gut-brain axis is involved in our mood?
Absolutely. So one of the very first papers on the gut-brain axis actually showed a direct effect of the gut microbiome on our stress levels. When I say ours, I actually mean mice, because like most of the research on the gut microbiome has been carried out in rodents. And most of the experimental design when it comes to studying interaction between the gut microbiome and our brain or our moods uses a specific model called the germ-free mouse model. And these are mice that are depleted of gut microbes. They don't have a gut microbiota. This is either by birth or by being treated with antibiotics that deplete the and kill all gut bacteria. And we see that germ-free mice have abnormal anxiety levels and abnormal HPA axis. The HPA axis refers to the hypothalamic pituitary adrenal axis, and it's the innate stress response system that is common to all mammals. And so, yeah, there's definitely a correlation between our gut microbes and our stress levels and our HPA axis. And that's one of the mechanisms through which this is one of the mechanisms that is common actually to both the gut microbiome and psychedelic drugs, because it's a target of both.
Oh, interesting. How do we know how psychedelics interact specifically with the HPA axis?
There have been some studies that have tried to answer this question, and results are still quite unclear. So, same with inflammation, actually, but even more for the HPA axis. Some studies suggest that certain psychedelic drugs increase HPA axis reactivity. Others suggest that it decreases it. So others suggest that it's a time and dose related response. So I think once again, we do need more research to find out the exact nature of this relationship.
And when you say increase versus decrease, does increase mean increased stress, or is that what does it mean to have increased HPA axis activation or activity?
So normally this refers to cortisol levels, but it varies depending on studies. Sometimes it can just refer to how reactive we are to stress stimuli. But yeah, the cortisol levels is usually one of the classical readouts.
Okay. And since we have you here, somebody with all this knowledge, I'm just going to ask some associated questions, because you also brought up that we do know something about how psychedelics affect the immune system. Is that correct?
That's right, yes. So once again, most of the literature suggests that psychedelics elicit a decrease in a lot of the inflammatory cytokines or inflammatory mediators that are usually associated with an immune response. If you're familiar with it, you might recognize interleukin-1 beta, also TNF alpha, CRP, which is an actual marker, a circulatory marker of inflammation. They all seem to be decreased by psychedelic drugs, although once again, there is not all research is agreeing on this topic, so we need definitely more research to evaluate whether this is a definite statement.
And has the research that we do have into that topic been done only on the serotonergic psychedelics?
No, my review paper focuses on serotonergic psychedelic for a reason, which is serotonergic psychedelics, as the name suggests, they're very related from a structural perspective. They're very close to serotonin. And serotonin is strictly related to gut physiology. So because we have such limited direct evidence of an effect of psychedelics on the gut microbiome, my review paper focused and draws on what we know about serotonin and its relationship to the gut microbiome. Also, another important fact to mention is that I'm sure all of your viewers know, but psychedelics, especially serotonergic psychedelics, they are agonists at the serotonin 2A receptors, which which means they bind to the receptor and they activate it. And so for me, it made sense to draw evidence from the interaction between the serotonergic system and the gut microbiome. And we have lots of evidence like that.
I absolutely love it. And just for the listeners who might not know, can you inform them which are the serotonergic psychedelics?
So these are also known as classic psychedelics. So we're talking about psilocybin, also all of the analogs like norbicistin that I mentioned earlier, DMT. Ayahuasca obviously has DMT as the active component. The non-classic psychedelics are things like ketamine, for example, that have also bind to serotonin receptors, but they also have additional mechanisms of action that include other neurotransmitter systems like glutamate, for example.
Are there any psychedelics, because I just don't have this off the top of my head, that don't interact, that we know don't interact at all with the serotonin receptor?
No, they all do. All psychedelic drugs react and bind to serotonin receptors. It's just a matter of whether that's the only or the primary target. But yeah, and actually, research suggests that the affinity of psychedelics to the serotonin 2A receptor is strictly correlated to the subjective effects. So the mystical experience and psychedelic trip that you get when you're taking psychedelic drugs.
Okay, thank you for that clarification. Now I'd love to get into basically the heart of the matter, which is the why is this important? So that comes down to the precision or personalized medicine that we discussed before. So if you could even just go into a little bit more than I briefly did in the intro about what personalized medicine is.
Okay, I love this topic because my background is neuroscience and my entire PhD, I focus on depression. And you're a clinician, so you know that for depression and a lot of other disorders, the prognosis is really a problem. And this is because depression is a very heterogeneous disease. It's very complex and it's very severe and debilitating, obviously. And because we still don't have clinical biomarkers for depression, which means we don't have a way to measure it quantitatively, it's very hard for clinicians to be able to tell what treatment will work for that particular patient. And so my entire PhD pretty much focused around identifying biomarkers that could predict response to antidepressant drugs. And this is what really ignited my interest and passion for precision medicine, because it really like identifying biomarkers that can tell, that can tell what patient will benefit from what particular treatment. Like it has incredible implications. And the gut microbiome is a specific great target for this, actually, because the gut microbiome is different in every person. It's very unique. And so every person carries this unique microbial signature that can potentially map out to all of the treatment options that we already have available. It's just a matter of understanding and identifying markers that can predict which patient will benefit from a specific drug. So predict treatment outcome, but also predict side effects, like you mentioned, and also trying to stratify the population in different clinical subgroups so that we can match, as I mentioned, existing drugs to the biology of the individual patient. So how is this relevant to psychedelics? Obviously, with psychedelics, just the way psychedelic assisted therapy, for example, is structured. It's a very costly procedure. It requires lots of time. It requires lots of practitioners giving a lot of time, a lot of effort to a single patient. And sometimes this doesn't really work out. So it's very important, specifically, even more so, I think, for psychedelic assisted therapy or psychotherapy, that we have some means to understand which patients are going to benefit from it and which patients are going to find some adverse reactions or side effects. And we already have ways to do this. Usually this is based on genetic information. For example, polymorphisms in the genes that are responsible for metabolism of these drugs. Depending on whether you're a fast metabolizer or a slow metabolizer, you may want to get different doses of psychedelics. There's also variability in the gene that encodes for the serotonin 2A receptor, which obviously also has implication because it's the target of psychedelics. And I think obviously this is very early days to be able to make any statement with regards to how the gut microbiome can be used for this purpose. But just based on how heterogeneous it is, it really has a lot of potential to predict treatment outcome, to stratify the populations that we're able to match different individuals with different psychedelic drugs, different time intervals or dosing, and also predict potential side effects. So this is the general idea behind personalized medicine applied to the gut microbiome and psychedelic drugs.
I love that. And if we could talk even just a little bit, and like you said, this is perhaps, years and years in the future where we'll have the ability to know how to do these matching things. But I always love to bring up things that people perhaps can use now. And so let's talk about some of the things that could go into precision medicine. One, like you said, is the gut microbiome. Two is just DNA, their regular DNA makeup. Some other things that I also think of are some of the ways that their brain works, right? Like some people have a
Absolutely.
preference for groups versus not groups. So you wouldn't want to put the person who prefers to be in a solo situation in a group therapy situation. So are there any other things like that that people can consider right now and how they may be not just following like, okay, everybody's signing up for this retreat or everybody's trying this certain type of ketamine. Let's go with the legal way. Some other things that people maybe want to consider when they're deciding on therapeutic options, even right now.
Yes, that's a very good point. I think from a research perspective, people are also really focusing on neurotypes, so neuroimaging signatures that are specific to the patient and can potentially predict either treatment outcome or help in identifying subgroups within the patient population. So that is definitely one way to go. From a practical perspective, it's maybe less accessible than a genetic screening or like sequencing the gut microbiome. In my PhD, this is not specifically related to psychedelics, but in my PhD, I found that specific plasma lipoproteins, so these are lipoproteins that are found in blood, could help discriminate patients that responded to two different pharmacological antidepressant strategies. So that's definitely an avenue to follow for research. But then, as you said, genetics, epigenetics, actually something I'm involved in at the moment is a study with ayahuasca. We're looking at basically this is a study on ayahuasca for PTSD and trauma-related disorders, and we've recruited participants with a history of trauma, and they're undergoing this ayahuasca retreat where we're collecting different biological measures. We're collecting blood, we're collecting urine, we're collecting fecal samples for microbiome analysis, as well as psychological questionnaires. And the idea there is to be able to eventually, once we collect enough data, to understand whether there's a match between the biological data and their outcome, their psychological outcome later on. So the idea there is really to try and identify some biomarkers that can be used clinically to predict outcomes to psychedelic therapy.
Very, very cool. Oh, I'm very excited to see when that paper comes out. I love that. It's what, when we have all these researchers out here doing these amazing things, and it's not possible, but you want to be like, can every single one of you give this long list of questions and take all the fecal samples and then take their DNA and check all. So we just have this like massive database of like figuring these things out. But we know that's not possible, but I'm very excited that you're doing what you're doing. And that leads into what in your paper, you discussed 3 specific implications for what we'll be finding through these studies, how that can impact the like psychedelic, personalized psychedelic assisted therapy in the future. So the first one you talked about is targeting the microbiome to modulate the effectiveness of psychedelic therapy. Can you discuss that a little for us?
Yeah, so that builds on the hypothesis that the gut microbiome shapes individual responses to psychedelic treatments. And so, if that is correct, if that is true, we could potentially target the microbiome, change the composition of the gut microbiome like we wanted to be able to maximize the effectiveness of these drugs. So that is what it refers to. Obviously, there is a lot of work to do before that becomes a possibility. We need to be able to understand and we need to know what microbes are responsible for metabolizing psychedelic drugs, for once. Also, it would help to understand the effect of psychedelics on the gut microbiome composition. And so this is like very early days, once again. But yeah, I'm very excited by that opportunity.
That's very cool. And the second of the things you suggested was with that information, can we predict response to psychedelic treatment from these microbial signatures? So what would that look like?
That would look like a bit like what we're doing at the moment. So collecting all of these baseline data. So this is data that we collect from the participants before they even take psychedelic drugs. And then after they take the drug, we look at their outcome. This could look like psychological questionnaires. Have they had any improvements from a psychological perspective on whatever their upset was? It could be side effects, adverse reactions. And so we collect all of this information and then we draw connections between the baseline data and this output. And the idea there is to be able to identify biomarkers, hopefully a panel of biomarkers that span different biological systems that are able to inform us on whether that participant will respond to psychedelics and how.
And this brings a question to mind because we're talking about psychedelics primarily, obviously, if we're talking the gut-brain axis, we're talking about psychedelics that are metabolized via the gut. Do you foresee that this also might lead to a situation where we're trying to decide oral ketamine versus intramuscular ketamine, and would the gut microbiome perhaps play a decision in that?
Absolutely. That's definitely it. And I think that refers to my third point in the review paper, which is exactly that. We can find subgroup of patients, and depending on their microbial signature or other types of biomarkers, we can start matching the patient to whatever the treatment. And when I say treatment, I don't only refer to which drug, but also which dosing. Which mode of intake, so whether it's administered orally or intravenously or intramuscular, and also time interval, potential augmentation therapy. These are all things that we could potentially one day figure out just based on baseline biomarkers.
Very, very cool. I'm very excited for this future. And I love all the research you've done and are doing. So I would love for you to tell people where they can find out more about you. Like I said, we'll link your paper in the show notes, but where can they find out about you and your research?
You can keep up with what I'm doing on my LinkedIn, my Twitter. I would say the best way to keep up with the research we're doing in relation to gut microbiome and psychedelics is to follow Onaya. Onaya is a charity that is doing a lot of work in the context of ayahuasca as alternative treatment for a lot of trauma-related, primarily brain disorders. And so if you're interested in this, please go check out the website. We do a lot of outreach as well. So I'm sure you'll have a good time scrolling through the posts there.
Very cool. Is that Onaya?
That's right.
Okay, beautiful. Giorgia, thank you so much for, first off, what you do, and secondly, coming on and sharing it with everyone today.
Thank you so much. It was great.
All right. For everybody else out there, until next time.